Okay, so always I view the research in three segments. One is the beautiful land, and a dragon came into the beautiful land. A sword we need to kill the dragon. Here, the beautiful land in my research is the human brain, which contains many neurons. These neurons, they are supported by various kinds of proteins. Among all the proteins, I wanted to study one protein called tau protein, because this is our dragon in my story. It leads to the Alzheimer's disease in unhealthy neurons. So what happens in the unhealthy neurons? In unhealthy neurons, this tau protein first forms droplet-like structures, and it allows certain kind of particles to enter inside the droplets. And then these droplets try to aggregate or form tangles, leading to the Alzheimer's disease. So we really need to make the sword to stop these tau aggregates. So the approach we make to make the sword is in 3 steps. Step 1, we form the droplets in the lab. Step 2 is we wanted to see which particles are getting invitation to enter inside the droplets, or which are the particles getting attracted towards the droplet. This is a very crucial step. So for that, we choose a technique called FCS technique, fluorescence correlation spectroscopy. So basically, this technique allows to distinguish between faster diffusion versus slower diffusion of the fluorescent particles. So our hero is ready, the droplets, our technique is ready. Now step 3 is we design various fluorescent particles with various properties, either change in the size or the charge, etc. And we introduce them into the droplets. And once we introduce them in the droplets through the FCS technique, we will see which is that one particle with that unique property getting attracted towards the droplet. So what we achieve from this is, we will know that property which is really needed to develop novel inhibitors for the Alzheimer's disease. So that is our sword, where we can kill the dragon, that is the tau aggregates, that is nothing but we can control the Alzheimer's disease. Thank you.